Welcome to video 5 of topic 6, human physiology. In this video, we're looking at neurons and synapses. So in this unit, the essential idea is that neurons transmit the message and synapses modulate the message. So basically meaning neurons pass the message along and synapses then push it across a space between the neurons, so the nerve cells as such. We'll obviously look into this much further. Our nature of science, cooperation and collaboration between groups of scientists, in this case biologists, are contributing to research into memory and learning. So there's lots and lots of research around this area, from reaction rates and age through to research into memory loss conditions such as Alzheimer's, things like that. So it's a really, really fascinating area of science to become involved in. Okay, so before we get going, this is just a little bit of revision from many years ago, okay? We're looking at the nervous systems. We've got two types of nervous system, all connected together, but we've got the central nervous system, which is the brain, spinal cord, okay? And then we've got what we call the peripheral nervous system, which is all the nerves, okay? Now the peripheral nervous system runs from or off of the central nervous system, so they work together. Now, as well as our nerves, we have what we call our receptors. Okay, so that's like the skin, eyes for sight, we've got taste, we've got smell, we've got hearing, so all different receptors that also are connected into our nervous system. Okay, and then just looking at the diagram on the right here, we've got all the different nerves as such. You're not expected to know them all, it's just having an understanding that they exist. Oh, go back. So here we've got a picture of what you would have looked at in years 8 and 9, a nerve cell, hopefully moving on to calling them a neuron. Okay, and we're going to look much more at them this lesson, or in this video. Okay, our understandings. So first up, neurons transmit electrical impulses. So the whole nervous system runs through these pulses of electricity, basically, that run through the neurons. Now we're going to look at how that actually occurs, okay, in a lot more detail. But knowing that charge runs from the cell body along the axon, and then from those terminal buttons that they're called there, the message then jumps across what we call a synapse to the next neuron. Okay, and that's how these messages are passed along. We're going to look in much more detail at that in this unit. Okay, so transmitting our electrical impulses. Let's look at that a bit closer. Now, we've got approximately 85 billion neurons in the human nervous system. They assist internal communications by transmitting nerve impulses. So we've got two systems of communication in our body. We've got the nervous system and the endocrine system, which is hormones, which we'll look at in the next video. But the nervous system works by transmitting what we call these nerve impulses, and they're actually an electrical signal, as that says. Now, when you think of an electrical signal, all we're thinking of is charge. Don't be bamboozled. An electrical current is just a movement of electrical charge, and we know that different ions, so atoms with a charge, carry this energy. So we're looking at the movement of these ions through the system, causing a bit of a charge imbalance, which is our electrical signal. Okay, so before we can start, we need to understand the structure of our neuron. They have a cell body, which has cytoplasm and nucleus, just like the cell body of all cells. However, they are specialized cells, and you would have looked at the fact that they're specialized cells when you're in year 8 and 9. They've got what we call dendrites. Okay, these are short branch nerve fibers, and you can see them. They're the little pokey bits out around the cell body. Now, these transmit nerve impulses in certain parts of the brain, spinal cord, from the receptors in your skin. Okay. Our nerve cell also has an axon. Okay, now these are elongated fibers. They make this long towel-like fiber. And they transmit these impulses from the extremities. So imagine from the end of the dendrites through down the axon to the axon terminal, when it then jumps across the synapse to the next cell. So if you're thinking about these actual nerve fibers, okay, they're cylindrical in shape. They have a plasma membrane enclosed in a narrow cytoplasm. Now, nerve fibers conduct impulses at a rate of approximately 1 meter per second. Alrighty, so you've got the idea of this neuron, and you've got the idea of these long nerve fibers, especially along that axon. Now, that axon is then covered in a myelin sheath, okay? Now, let's have a look at what this actually means. So your next understanding is the myelination of nerve fibers allows for saltatory conduction. Alrighty, so we've got two things there. We've got a process of myelination. We need to look into what that is. And then we need to look at what saltatory conduction is to understand why myelination allows it. So the myelinated nerve fibers, let's look a bit closer. And this is where we're looking along the axon. Okay, so that nerve fiber runs down the center, and then you see these it's called a myelin sheath that surrounds it and kind of goes along in bobbles as such. So, as we said, myelin coats our nerve fibers. It consists of many phospholipid bilayers, so layers and layers of phospholipids around our nerve fiber. We have these Schwann cells, and they deposit that myelin by growing around the nerve cell. So a bit like this, you've got your nerve fiber in the middle, and then the Schwann cell sheaths around, and that grows the myelin. So here, if you have a look at this, you've got your Schwann cell, and then that produces the myelin to grow around the nerve fiber. So these can be about 20 layers thick of our phospholipid bilayer before the Schwann cell stops growing. A gap between the deposits of Schwann cells is known as the node of Ranvier. So this is our node of Ranvier, okay? And that's like a gap. So this is one Schwann cell. So this is one Schwann cell with all its layers of myelin, and then you have the gap, which is the node of Ranvier, and then the next Schwann cell and all its layers. Okay, so we've looked at myelination, and those Schwann cells causing those layers of phospholipid around the axon, the nerve fiber in the axon. Now we need to know what saltatory conduction is. Okay, so this is where nerve impulses jump from one node of Ranvier to the next. So we've got our nodes of Ranvier here and here, and we've got this conduction between them. This is quicker than a continuous transmission along unmyelinated nerve fibers. So if you just had nerve fibers running along there with no myelination, it would be a slower transmission rather than here where we've got impulses jumping from one node of Ranvier to the next, okay? Now the speed for myelinated nerve fibers is 100 meters per second. Now just now we was looking at 1 meter per second. Okay, so once we've got that myelination, we've increased the speed by 100%. Okay, let's have a look. So propagation, that's a word you're going to hear quite a lot. That means that making of the impulse. So if you have a look at the picture, you can see sodium ions are entering so this is the nerve fibre along here, okay? Your cytosol, you're in the nerve fibre. So you've got sodium ions entering here. You've got your myelinated sheath here, so you've got, these are your nodes of Ranvier in between. All right, so sodium ions enter. You've got a different in energy potential. You've got more sodium ions here than here. So the sodium ions move along, okay? And that is causing our current to move along, right? That movement, diffusion of the sodium ions, causes the movement of these conduction, so the impulse. Okay, so we see that sodium ions need to get into the nerve fibre to cause that movement of current along the nerve fibre. Guys, I want you to think back to topic 1 where we had a descriptor about sodium potassium ions across the axon. This is where we finally look at that, okay? So earlier in the course I said we'll leave that, we'll come back to it. Well, finally at the end of the course we're coming back to it. So neurons pump sodium ions and potassium ions across their membranes to generate what we call the resting potential. Now the potential is the movement of current, and we've got different potentials, whether we've got a lot of current, not a lot of current, that we'll talk about. But first of all, we need to get those sodium ions or the potassium ions into the nerve fibre, into those cells, okay? And using our sodium potassium pump, that's how that's going to happen. Let's have a look a bit further at this. Okay, so the transfer of sodium and potassium ions across the membrane require a sodium potassium pump. It is an active pump. It requires energy, remember. Sodium ions are pumped out, potassium ions are pumped in. An unequal number of ions are transported across the membrane. We have 3 sodium ions being pumped out, 2 sodium ions being pumped in. We then get a concentration gradient occurring for both ions. This is our resting potential. So we've got a difference in the number of sodium and potassium ions, and we need this. Now this is actually a step before what we just looked at, okay, because we've now got less sodium ions inside the nerve cells, okay. It isn't equal, it isn't at equilibrium. We've got our resting potential. So our resting potential, we've got it's in a non-transmitting neuron, so it's not at that stage transmitting, but we have a potential difference. We've got voltage, okay, due to the unbalance of charges across the membranes. We are ready to transmit, but we're not yet transmitting. Now we get an action potential when we have what we call depolarization and repolarization of the neuron. So the neuron. This is as the current passes along the neuron. So depolarization is where we go from a negative charge to a positive charge. Repolarization is positive charge to negative charge. So our resting potential, okay, our neuron membranes are also more permeable to potassium ions compared to sodium ions. So that also causes this difference in charge. So potassium ions leak back across the membranes quicker than our sodium ions. There are also proteins within the nerve fibres which are negatively charged. Now this also adds to this charge imbalance, okay? And whilst we've got this charge imbalance, nature wants an equilibrium, Okay, so with the charge imbalance, it wants to get back to an equilibrium, and trying to get back to that equilibrium, you get that nerve impulse. So you see, we were at this stage, okay? We was talking about the potassium being pumped out. So we'll start here, okay? We've got potassium now coming in. So we've had that resting potential where the resting potential where the sodium was higher on the outside. Now the sodium's coming in. As the sodium comes in, it diffuses along, the current passes along the axon, so the nerve fiber passing along here, okay, because the sodium's like trying to reach an equilibrium, so it comes into the axon fiber. And then, because this area is higher charged than this area, it passes along. And as the sodium passes along, the potassium then is moving out and again making that potential difference. And that's our depolarize and repolarization. Okay, so action potential. Let's have a look at depolarization. So due to the opening of the sodium channels, sodium ions diffuse into the neuron. So we've got this, remember, all the sodium was out at that stage. Now we're letting it in, like at the start of that diagram before. This reverses the charge imbalance across the membrane. So inside the cell now becomes positive compared to outside the cell. And that moves along the axon. Behind that, those sodium channels shut, the potassium channels open, potassium ions diffuse out of the neuron, recharging the neuron. So outside of the cell then loses that charge. So these nerve impulses are just the movement of these action potentials along the axons of the neuron. So the movement of those changes in current as such, or in charge, that's what our nerve impulse is. You can see that here, that area of depolarization, because of the sodium ions entering the axon, that moves along, you repolarize after. This resting is where that sodium's being pumped out, okay, so it makes that charge imbalance. Sodium enters here to try and rebalance it, and then that passes along. After the sodium, we've got the repolarization, and then the resting comes along again. So it's just this continuous cycle, and this pulse of depolarized ions as such, or current, moves along the axon. And we have what we call, when we look at the membrane potential here, you can see how that impulse is measured, okay? So our threshold potential's there. So if we have a look, we've got our resting potential, and then we have the depolarization occurring where our sodium ions are entering the cell. And then once we've hit that, we then repolarize, and along comes our resting potential again. This is a diagram showing that, okay? Watch your little red bit as it skips along, then it goes back again. Okay, so skips along. All right, local currents, and that's what, if you imagine in each neuron, so each axon, it's a local current that's occurring. So action potentials along an axon is due to the movement of these sodium ions, as we've said. That depolarization occurs when sodium ions diffuse into the axon via those sodium channels. So we've got that charge imbalance during the resting potential, and then as depolarization occurs, those sodium channels open and the sodium comes in. Okay, now this isn't your sodium potassium pumps, okay, that's when they're pumping the sodium out, okay, against the concentration gradient. So at this stage, concentration of sodium ions across the axons is decreasing. The concentration of sodium ions inside the axon is increasing. So and this is just by simple diffusion through the sodium channels. That is a passive process, remember, through the sodium channels. So our sodium is coming in. That's our action potential. Okay, now the depolarized part of the axon has a different sodium concentration to its neighboring parts, and this causes the sodium ions to diffuse between these areas, so to move along. Okay, so these sodium ions are going to move from here to here just by simple diffusion, but by doing that, they carry this current with them, which is the nerve impulse, which is the action potential. Again, this graph is just showing this. Okay, you've got your active potential in the middle because your sodium's entering. So our local currents occur due to the difference in sodium concentration inside and outside the cell. They reduce the concentration gradient in the area of the axon not yet depolarized. That causes this wave of depolarization and repolarization along our neuron, which is the nerve impulse. Alrighty, so if we've worked out how our nerve impulse moves along the axon, next up we get to the end of the neuron and we need to cross a synapse. Okay, so a synapse is the junction or the space between two neurons. Okay, so the impulse won't pass across like it's been moving along the axon or along the cell. It now needs a different way to get across that gap between the neurons. Okay, so as I said, synapses is a space in between the cells where two cells meet. It can also be where a receptor and a nerve cell meet. To get the impulse across the synapse, we need to use these things called neurotransmitters. And that's because an electrical impulse will not move across what we call the synaptic cleft, which is that space. Okay, so when the presynaptic neurons are depolarized, they release what we call a neurotransmitter into the synapse. Okay, so that action potential has got to this point, okay, and then they release these neurotransmitters across the synapse. So we need a stimulus. Something needs to stimulate the nerve impulse. That propagates the nerve impulse along the nerve cell. It arrives at the presynaptic neuron. So it arrives at the end of the neuron next to the synapse. The membrane depolarizes because those sodium ions have come along. Now we get calcium coming along. So calcium ions diffuse through the membrane channels. So we've got calcium ions here. This causes vesicles containing neurotransmitters to move to the membrane. So this change in charge causes these vesicles to move towards the membrane on the end here. They then fuse with the membrane and out. So inside these vesicles are what we call these neurotransmitters. Once the vesicles fuses with the membrane, it releases those neurotransmitters into what we call the synaptic cleft. And as you can see by this picture, I think you'll be able to tell what's going on here, because these are the neurotransmitters and they are attaching to receptors. So via exocytosis, neurotransmitters are released into the synaptic cleft. They diffuse across the cleft and then they bind to receptors on the next neuron. So this is the next neuron. Okay, so now we're going for postsynaptic neurons. So we're into the other neuron now on the other side. So they have recognition of binding of the neurotransmitters, and this causes sodium channels to close to the receptor site. Sodium channels close to the receptor site to open. Sorry, that didn't make any sense how I just read that. So now sodium ions diffuse down the concentration gradient into the postsynaptic cell, so into the postsynaptic neuron. The membrane reaches its threshold potential, and our synaptic transmission impulse is generated. So an action potential is then triggered. We've got sodium coming in. We're going to propagate that along the neuron. Our action potential is going to move along the neuron like we've spoke about already. And our nerve impulse is formed. So a nerve impulse is only initiated if the threshold potential is reached. We've got lots of different potentials, haven't we? So this is our threshold potential. So we need to get enough sodium ions to reach this threshold point to allow the action potential to form. So at the threshold potential, the sodium channels open. This causes depolarisation. The diffusion of sodium ions into the cell increases the membrane potential, causing more sodium channels to open. Action potential is then initiated by the threshold potential being reached. Alrighty, so now let's move on to our applications and skills for this unit. So our first application is understanding that the secretion and reabsorption of acetylcholine by neurons at the synapses. So this is really interesting. This is looking at those neurotransmitters and this chemical or substance called acetylcholine. Now this is used as a neurotransmitter. It's produced in the presynaptic neuron from choline and acetyl groups. Endocytosis forms vesicles that contain the chemical, and it's released into the synaptic cleft during transmission. It binds to the receptors on the postsynaptic neuron, and then an enzyme, acetylcholinesterase, breaks it down to choline and acetyl group again, and then it's taken back round into the presynaptic neuron. Okay, now this is a great application. This is looking at how nerve impulse transmission is blocked by certain pesticides, okay, at the acetylcholine receptors. So this is how some pesticides work. So let's have a look at this. What's happening is we've got a blocking of synaptic transmission, okay. It happens at these cholinergic synapses, which we've looked at, okay. It's happening in insects, and it's by binding of these neonicotinoid pesticides, okay. So these pesticides bind to the receptors that would usually pick up the neurotransmitters, okay, those receptors that we've already looked at. So the pesticide binds to the receptors, so the nerve impulse can't be passed on. Therefore, no impulses, no nervous impulses, the nervous system of the insect then fails. So another skill is that you can look at the graphs that show the different potentials in a nerve impulse, okay, so that you can say, well, at this point we're in the resting potential, sodium ions are entering our axon here until the threshold potential is reached, then those sodium channels open, we've got the depolarisation that then occurs until we reach action potential. When we reach action potential, that pulse moves on, and that area behind where the current has just been, that high amount of sodium ions has just been, then those potassium ions start to leave. We get back to our resting state. We actually fall below our resting state first of all until that equilibrium is found again. That is all for our video on 6.5. I'm looking forward to seeing you in our final video, 6.6.